Hey everyone, Mr. Richardson here. Just wanted to make this little video to talk to you guys through the process of chemical evolution. So we talked a little bit about in class that this idea of chemical evolution is the formation of complex molecules necessary for life are formed from simpler molecules, and then those simpler molecules are even further more simple atoms. This happened billions and millions of billions of years ago. And so this video is kind of a walkthrough of how we go from simple building block of atoms to build small molecules, to make bigger molecules, to make macromolecules or big molecules. More specifically, what we're going to be doing is using atoms to make small molecules to make amino acids, and eventually use amino acids together to make proteins. I do want to emphasize, though, before I continue, that even though this video is about this whole unit is about the basics of life and relating to that, nothing in this video is living. Amino acids aren't alive, proteins aren't alive, they're just fundamental for all life to occur. So during this video, I'm going to kind of walk you through that, the molecule kits, the process of doing that and highlighting some of the major things. So I recommend during this video pausing it. If you need to correct your notebook or draw some colors, it will help. Also have those follow-up questions. I'll make sure to try and highlight those through as I go through it, so you might want to pause it and maybe rewind it if you think you heard something I talked about. So good luck. Thanks. So what we have here are the basic atoms that we'll be using, the four atoms. From left to right, we have first the hydrogen atom, which can bond one time to one other atom. We have next the oxygen atom, which can bond to two different atoms. The nitrogen can bond to three atoms. And finally, carbon can bond to four different atoms, making it very versatile. Here you can see we've created our first set of small molecules by bonding some of these basic atoms together. On the far left, we have hydrogen gas with two hydrogens, then the famous water, H2O, one oxygen, two hydrogens. Then we have a black carbon with four hydrogens, that's methane. Then we have an ammonia, which is that red nitrogen with three hydrogens. And then the last one's a carbon dioxide. Now that one's a little bit different because it is a double bond between two oxygen atoms. That carbon double bonds twice to two different oxygens. That's why you see two little white strips between the two of them. Alright, so to start building our more complex molecules, we're going to start with a methane and we're going to remove three of the hydrogens. And this is what the central carbon, now that has three extra places to bond, is going to be what we call our central carbon. This is going to be the centerpiece of the amino acid that we're going to be building. Everything's going to be added on to this piece as we go forward. The first functional group that we're going to add onto that central carbon is what we call the amine group. And it's formed from an ammonia. And so what we're going to do is we're going to take the NH3, the ammonia, we're going to remove one of the hydrogens, we're going to set it aside because we will be using it later, and we're going to connect it to the central atom. So this first step is pretty straightforward. So now we have the central carbon on the left connected to the amine group. Alright, this is where things can start getting a little tricky, and it has to do with the carboxylic group, our next functional group attached to that central carbon. So you can see it on the left. Right now the pen is pointing to the central carbon. On the right side is the amine group. To the left, we used a carbon dioxide. We broke one of the hydrogen, or the double bonds, and attached a hydrogen to the oxygen, and then connected that carbon and the carbon dioxide to the central carbon. That is our carboxyl group, the carboxylic acid. This one's a little bit tricky, but right now what we have here is very important. What we have right here is the basis of every single amino acid. Every single amino acid has a central carbon, of course, and then the same two groups, an amine group and a carboxylic group. And there's one empty spot. There are various types of amino acids. There's proline, leucine, valine. There's over 20 kinds of amino acids, and every single one of them has a carboxylic group and an amine group attached to a central carbon. The difference between all of them is the very last part, and it's actually what my pen is specifically pointing to, the last place on the central carbon, and that is called the radical. The radical is very important because this is what determines which specific amino acid we are going to have in this situation. Okay, for this specific amino acid, you can see again the amino acid or the amine group, the carboxylic group, but I added a hydrogen to the location which we call the radical. That makes this a glycine. Glycine is the amino acid that has a hydrogen in the radical position. So what we're going to do now is, now that we have our finished amino acid, we are going to try and create another amino acid so that we can combine them to eventually make our protein. Okay, so here we have two different amino acids. The one on the left is the one we've been working on this whole time. That's glycine. You can tell because the bottom part of the central carbon is a hydrogen. The one on the right has the amine group on the far right, a carboxylic group on the left, and on the bottom part, instead of a hydrogen like glycine, we have a CH3, another carbon with three hydrogens attached to it. That makes this, since the radical is different, it identifies this amino acid as a alanine. Okay, so now what we're going to do is we're going to have a chemical reaction to combine these two amino acids to create a larger macromolecule. More specifically, two amino acids will make a protein. So in this case here, what we have set up is we have on the glycine on the left, I've removed a hydrogen from the amine group, and on the alanine on the right, I've removed an OH from the carboxylic group. And we're going to use these two now open locations to bond them together, forming a peptide bond. Alright, so I've combined those two amino acids together to form my protein, my big protein. And right where my pen is pointing is that location where those two amino acids chemically bonded together. Since these are amino acids, that that bond is specifically called a peptide bond. Peptide bonds are a type of chemical bond that hold together amino acids. But I had taken off a hydrogen from the amine group and a OH from the carboxylic group. If we combine them, at the top there you see when we combine the H and the OH, we actually end up with water. This is actually an example of something called a dehydration synthesis. It's a creation of a protein and water from the reaction, dehydration, because you're losing water in the reaction. Okay, so to recap here, we took a glycine, which is an amino acid, an alanine, another amino acid, and created a protein and water, which I said is a type of, this is called a dehydration synthesis reaction because we're losing water. You're dehydrated, you're losing water in this reaction. All right, so in this case here, we take, you can see the little circles I drew there with the lines between them. I took an amino acid to make a protein, but most proteins are actually many amino acids bonded together. Some hundreds, some thousands, some hundreds of thousands. But each one of them is held together with a peptide bond. So an amino acid connected to amino acid, connected to amino acid. We get this long chain, and that's what forms the protein. But amino acids are a very specific type of reaction. Amino acids are just a type of what we call a monomer. A monomer is a much more broad term, not as specific as amino acid. So actually what we really did here, since we're actually talking about all four macromolecules, proteins, nucleic acids, lipids, and carbohydrates, They all are big molecules formed by smaller things, just like proteins are formed from smaller amino acids. But those smaller things are called monomers. And you take a monomer and sometimes many, many more monomers to create a polymer. Mono meaning one or single, poly meaning many. So you take a lot of little ones and combine them together. So, in kind of in a more general term here, we can take a a monomer, bond it with a monomer, bond it with another one, and another one, and another one, using chemical bonds to form a polymer. And the polymer is could be, like I said, protein. It could be a nucleic acid. It could be a lipid, or it could be a carbohydrate. And each one of that monomer will be a little bit different. For example, for nucleic acids, the monomer is not amino acids, but nucleotides. But we'll get more into that as we start talking about macromolecules.